Hello, I'm Will. Welcome to ResearchPod. Understanding the tiniest scales and interactions of why molecules act one way or the other within a cell is so far removed from a whole-body view of health that when you hear phrases like nanoparticle drug delivery, you'd be forgiven for the shock of being reminded that everything, even you, is just atoms all the way down. Professor David Punihaole at the University of Vermont leads a research team working with materials and medicines at that molecular scale, combining cutting-edge spectroscopic techniques and microscopic views to investigate, and have action on, the fundamental chemistry behind all our macro-scale health. Dr. David Punihaole, hello.
Hi, how's it going?
I'm very well. Could you tell us a bit about yourself, your background, and your research, and what brings us here today?
So my name is David Punihaole, and I am an assistant professor of chemistry at the University of Vermont. My research group is relatively new. I came to Vermont in August of 2020 during the height of COVID. I've been setting up my lab since then. My research group is interested in developing what we call chemical imaging tools that can be used to not only visualize the location of molecules, for example, but also can be used to look at their structural dynamics and interactions, to learn something about how these molecules behave chemically, which plays an important role in biological functions. And this research interest really stems from my background. I received my bachelor's of science in molecular biology and my PhD in molecular biophysics and structural biology, both from the University of Pittsburgh. And as I said, my research interests stem from my background, really trying to understand the molecular behavior of proteins and DNA and other types of biologically important macromolecules.
You say there's anything, philosophical that you find in connecting the whole human outcomes to these atomic interactions and atomic changes, kind of the very different scales being personal when played out at large?
Yeah, so I think back to a quote from the great physicist and Nobel Prize winner, Richard Feynman, who said that if you want to understand, I'm going to paraphrase him, but he essentially said that if you want to understand biology, be it the molecular basis for human diseases or just the fundamental workings of life, you have to recognize the fact that all biological molecules are composed of atoms, and that it's the wigglings and jigglings of those atoms that give rise to the functions of proteins and DNA, RNA, and all sorts of other types of biomolecules. So I think trying to understand those wigglings and jigglings of those atoms is extremely important to develop a holistic bottom-up view of how life works.
Let's come back to the Punihaole lab, which you mentioned there, being set up in the middle of 2020, a nice, calm, relaxing time for everyone involved. Can you walk us through a little bit of who's involved, what are they working on, and from there we can get into kind of the ongoing research that's coming out of it?
Yes, so my research group now currently we have 4 graduate students. My 2 senior graduate students are Maddie and Russell, who are both analytical chemistry PhD students. They are just finishing their 3rd year of studies. I have a 2nd year student, Lauren, who's a physical chemistry PhD student. And then my newest student is Shakira, who is also an analytical chemistry student, and she just is finishing up her 1st year. Maddie and Russell really built the lab. And this was very challenging to do during COVID because they did not come in with any knowledge of lasers or optics or anything like that. And it was very difficult to train students with social distancing requirements during COVID. But they are very natural at working in the lab. They're great scientists. And in the case of Maddie, she is working on trying to develop tools using a technique that our group specializes in called Raman spectroscopy. She's trying to develop Raman spectroscopic tools to study the structure of protein aggregates associated with Alzheimer's disease. Russell is trying to develop similar Raman-based tools to study the structural dynamics of polymers and other types of nanoparticle systems that can be used to deliver genes and drugs into cells. Lauren and Shakira are working together on a project where we're trying to develop imaging probes that can be used to not only visualize where the molecules are, but actually visualize or sense local chemical environments that the molecules are experiencing.
I can imagine it's been a busy couple of years for bioscience generally, but specifically looking at chemical engineered molecules sensing cellular environments. Would you say that this has been something that you've seen from the kind of hands-on perspective, or has it all just been happening and I've not noticed?
No, I would say you're right, at least certainly in this field that I'm in with Raman imaging. So, historically, people have been using fluorescent probes to tag proteins, for example, and these fluorescent molecules will emit light that we can then use to visualize where molecules are located in a cell, for example, with a microscope. So that work's been going on for several decades now. I think what's new is the realization that, we can do more than just use a microscope to visualize where things are located. We can actually use these things to visualize the interactions and the structural dynamics of how those molecules actually are carrying out their biological functions. And so there's been work now in the fluorescence field to do this, but also since I'd say the past decade, there's been increasing recognition in my field that it's a good thing to develop probes that can be used in conjunction with Raman microscopy to sense chemical dynamics as well. And so I would really say it seems like since 2020, several research groups have kind of all converged on that focus in my field.
All of that wiggling and jiggling that you mentioned.
Mm-hmm.
Well, I've got to say, the Punihaole website is a very useful resource for getting me up to speed with all of the content that's on there and all of the breakdowns. It's got lots of summaries, so if anyone listening at home wants to have a quick dive into the research that you're doing, it's a very good place to start. For some of the more in-depth details, we can crack on to talking about some of the resolutions and scales of what you're working with here, the magnification and microscopy involved there. Is there any useful metaphors or comparisons that anyone listening at home might be able to think, that comparing the Empire State Building to a grain of rice, that kind of thing?
So a cell, a biological cell, is somewhere on the order of a few tens of micrometers. That's about as thick as a human hair, for example. Molecules are, over an order of magnitude smaller than that. We're talking about length scales that are in the order of 10 to the minus 9th, 10 to the minus 10th meters. So very small scales, spatial scales that we're talking about. These are the kinds of interactions the kinds of length scales that we're trying to tease structural dynamics and interactions out of. If you take a conventional microscope, say in a high school classroom, and you look at some biological cells, you're using visible light to be able to see those cells, right? And so the microscope has a resolving power. Given the wavelengths of light that you're using to see those cells, the microscope has an ability to resolve objects on the order of about 200 nanometers, right? So molecules are even smaller than that, still a factor of 100, over 100 smaller than that.
Those molecules that turn into human conditions, into human cells, into human disease as well, to kind of ricochet back and forth between the very tiny and the very relatable human scale of things. Could we talk a bit about how some of the research happening at your lab relates to neurodegeneration, Alzheimer's, how you are connecting the chemical and physics research to the human outcomes?
Yes. So one of the research interests my group has is trying to understand the molecular basis for a host of different types of neurodegenerative disorders that seem to be linked to protein aggregation. The classic example of this is Alzheimer's disease, where there's a protein fragment that's called A beta, and this fragment will aggregate abnormally and form these large filamentous aggregates that are known as amyloid fibrils. These amyloid fibrils will then compact together to form plaques. And it's these plaques that if you do an autopsy on an Alzheimer patient's brain, you'll find that they're riddled with these protein plaques. And it's thought that there's a link somehow between this abnormal aggregation of this A beta peptide and Alzheimer's. What that link is, we don't know, but there's certainly some compelling circumstantial evidence suggesting that it's there. So, for example, you can do autopsies on different Alzheimer patients' brains, and these different Alzheimer's patients may have different clinical subtypes of the disease. And what you find then, if you look at the molecular structure of the fibril aggregates, you find that there's different structural types of these aggregates in the brain. And so that is one of those types of circumstantial lines of evidence that suggests there's a link between the structures of these different aggregates and potentially clinical phenotypes. It's also known that these aggregates can cause oxidative stress in cells, and that's one of the mechanisms by which these aggregates may end up killing neurons in the brain, and that then in turn would lead to neurodegeneration. So our group, studying these aggregates is very difficult, and historically it's been very difficult to do. There's only a few techniques available to study those aggregates, but all of them are done. All those techniques require that you study the structures of these aggregates outside of their biological environment, so outside of the native brain tissue environment that they're found in. And this can have a variety of problems associated with it. Notably, it's unclear whether the structures that scientists are studying in test tube environments actually are the same as those in the brain. It's also unclear that if you try to purify these aggregates out from the brain to study them, that is, it's unclear if that purification process can actually perturb the structures. And so what you're studying in the test tube environment is not necessarily what you're studying, what's clinically relevant in brain tissue. So that, was a big motivation for my group, where we're trying to develop tools that are structurally sensitive that can be used to study protein structure. And we'd like to develop those tools so that they're as non-invasive as possible, where we can actually use them to directly study these proteins and their aggregates in cells, in brain tissue, and things like that. So that's the ultimate motivation for our work, because our thought is if we can understand the structures of these different aggregates, we could potentially link the structures of these aggregates to different clinical phenotypes you observe in these diseases. We can understand really the structural basis for the different toxic activities of these aggregates in the brain.
Mm-hmm. And is that something that you see as being a fruitful avenue of research, or are you just getting started? Is there anything that's come out of that so far that a peer or another professional might be able to read more about, or early days still?
So the big picture idea that I described, we are just getting started. I do think it's a fruitful avenue because again, the amyloid field has been stuck on trying to prove that there's a direct link between the structure of these different types of aggregates and toxicity. And this field is there's been debates raging in this field for the past 2 decades. Some groups argue that it's not these large plaques that are causing the issues. It's actually smaller precursor aggregates that aberrantly interact with cells and causes cell death. And so, for almost 2 decades, it seems like there's been multiple different camps arguing about these things. And all of these questions, I think, could be solved if we had tools to just be able to directly study these aggregates in their native biological environment. It's high risk, but I think it's worth the payoff. It's going to finally resolve some of the big questions in the field. We have been working towards the first steps towards addressing these big questions, right? So, recently my group developed a technique where we can use Raman spectroscopy to measure select structural parameters in these fibril aggregates, things like different bond angles, for example. And we can then actually use these structural parameters that we measure as constraints in computational simulations. We actually then we take the experimental parameters we measure, we feed that into a simulation where we constrain the structure of our protein aggregate. We run that simulation and we get a set of structures that resemble our experimental data. And that's how we actually visualize the structures of these different aggregates. And we're hoping we can at least translate some of that methodology in the near future towards imaging applications.
Now, we've mentioned it a couple of times, but I think it's worth going over some of the concepts and the experimental setups behind the technology here of what is Raman spectroscopy and what that data could hold in the future.
So Raman spectroscopy, it's a very old technique, actually. I think it dates back to the 1920s. It was discovered by Sir C.V. Raman, who was an Indian scientist, and it's a light scattering method. The idea is you use laser light, typically, to excite scattering in molecules. Now, most molecules are going to scatter the laser light, so there's no change in energy between the incident laser light you use and the scattered light that you see. So if you have a green laser, you're going to see green scattered light get emitted from the sample. This type of scattering where there's no change in energy between the incident and the scattered light is called Rayleigh scattering. And it's actually the reason why the sky is blue, because there's a wavelength dependence to this light. The more blue the light is, the more efficient the scattering process. So the reason why the sky is blue is sunlight comes in to the atmosphere. You have nitrogen molecules in the atmosphere, and they can scatter the blue light much more efficiently than they can scatter the red light. And so what we see on Earth on a sunny day is blue light due to that process. Now, a little bit of that light, maybe 1 in a million photons, is going to get shifted in energy when the light is scattered. And so what's happening there is your incident photons interact with your molecules, and they drive vibrational motions in the molecules. And so as a result, energy is being used from the light to excite motions, those wigglings and jigglings of those atoms that we talked about earlier. As a result of that exchange of energy, the emitted scattered light is going to be shifted in energy. And so that's going to manifest as a change in the frequency of the light that we measure. So this is the Raman scattering process. And because our technique probes molecular vibrations, we can actually use those vibrations to learn about how the molecules are interacting with their local chemical environment. Because molecules will change their structure as a result of these interactions, and that will cause them to scatter the light differently. So this technique's very old. As I said, it dates back to the late 1920s, but historically it was very challenging to do. And that was because the technology really wasn't there to make this an easy type of experiment to do. So it really wasn't until the development of the laser that Raman spectroscopy became more popular. So it was discovered in the 1920s, but the laser was invented in the early 1960s. So it took several decades before lasers were invented. And the reason why lasers were so important was because they can emit much stronger light that can be used to excite the Raman scattering because it's such a weak process. And so as a technique to study biological molecules, you can use a laser to excite the vibrations of molecules like proteins or polymers. And then you can collect that scattered light and send that scattered light into a device called a spectrometer. And what the spectrometer does is it separates that light into its different color components, and you can then image that onto a camera. And that's how you measure what we call a Raman spectrum. And that technique works. It's still a very slow and painful process sometimes to do some of these experiments. One of the problems with conventional Raman methods is that a lot of biological systems, including cells, naturally fluoresce. So they emit fluorescent photons. And that fluorescence is rather strong compared to the Raman scattered light. And so you often will detect both the natural fluorescence of the cell as well as the scattered light from the Raman photons. And that can complicate and overwhelm your Raman signals. It makes it very challenging to do. And it's because of issues like that you can't do live cell imaging with traditional Raman methods. And that's why for a long time, biologists really did not want to use this technique. However, around 2010, the field started to change. And that was because the technology and detection systems had been developed so that you could do something called stimulated Raman scattering. So the idea here is you don't just use one laser to excite the scattering, you use two laser beams. And you tune the wavelengths of these two laser beams to particular vibrational resonances of the molecule. And you can then stimulate the emission of the scattering process. This has made it now feasible to do live cell, real-time video rate imaging of biological cells. And so that's now something that my group is moving into to further develop this technique. We've actually been constructing a simulated Raman microscope, and we hope in the next year or so that we can get our first live cell data from it.
Well, another avenue of research that the Punihaole website spells out quite neatly is the drug delivery avenue of research. Stuff about a quinine copolymer as an example of your work that leapt out to me. So I'm wondering if we could kind of hop from the post-mortem cells to the live imaging of cells and how the Raman spectroscopy and the amount of data you can get out of the scans that happen there are informing drug development and research.
Yeah, so this work really stems from my time as a postdoctoral researcher at the University of Minnesota. At Minnesota, there was a great cohort of researchers who were trying to develop alternative methods to deliver drugs and therapeutic nucleic acids to cells for cancer gene therapy type applications. So currently the gold standard method is to use viruses to deliver genetic cargo to cells to do gene therapy. Viruses are great because they've essentially evolved to do that task. But as you and the viewers will know, like viruses have a major drawback in the fact that they can elicit immune responses in patients. And actually, there's been a number of high-profile cases in gene therapy trials where patients have actually died as a result of using viral vectors for gene therapy. Patients have also suffered other complications. They've developed carcinomas, sarcomas, and other health issues. So viruses, as good as they are at efficiently delivering their cargo to cells to enable efficient gene therapy,
They are still viruses.
They are still viruses. And so the health risks are something you have to seriously weigh as a clinician when you're conducting one of these therapy trials. So, researchers at Minnesota and all across the world have been trying to develop alternative, safer vectors for delivering the genes to cells. And polymers are one such delivery vector. So many people, I think, are probably familiar with polymers. It's, polymers can comprise the materials that coat your nonstick pan, rubber from a tire, that's a form of a polymer. They are basically just long chains of molecules that are linked together to form a larger system that we call a macromolecule. But you can have polymers that are safe to use on cells, that are biodegradable. And these types of polymers seem to be really useful for gene therapy and drug delivery applications. The problem, despite their potential advantages for being safer, is that they are nowhere near as efficient as viruses at delivering their cargo. And a big reason why is because we don't understand how they actually work. So there's a lot of research groups trying to understand exactly how do these polymers interact with cells? How do they even get to the cell? so that they can get internalized and then deliver their cargo to the appropriate compartment in the cell. There's a lot of unknowns there. And part of what makes it so hard to study the mechanisms by which these polymers carry out their gene and drug delivery functions is we just don't have good tools to study these polymers. So I saw an opportunity here to try to make a difference with Raman spectroscopy. The system that we initially worked with was, I had a collaborator, Teresa Reinecke, and her research group had developed these polymers that contain quinine. Quinine is a molecule that is found in tonic water, for example. It's the oldest drug known in the Western world. It was used to treat malaria. So it's very biocompatible. And it turned out that incorporating quinine into their polymers really enhanced the gene delivery efficiencies of these polymers. And the big question was why? So we weren't sure we could actually answer this question, but we felt that we could use our tools to understand a little bit of how these polymers worked. And the first question we asked was, how do these polymers, where do these polymers deliver their cargo in cells? Can we use our Raman spectroscopic tools to actually track the cargo release in the cells? So it turned out the Raman signals that we found for quinine were very sensitive to probing how quinine interacts with DNA. And we could see characteristic shifts in the scattering frequencies of these quinine signals to figure out the different ways that quinine could essentially interact with the DNA. There's a different types of what are called non-covalent interactions that molecules can engage in, and we could actually figure that out with quinine. So we then use this signal to actually monitor in cells where the cargo was being released. And so we published a paper in 2020 on this with Teresa Reinecke's group, where we showed that we could actually visualize these non-covalent interactions directly in the cell for the first time using an optical microscope. So this was a really powerful advancement for the microscopy field.
Well, I mean, from everything that we've talked about, it really seems like your research sits at the junction not just between biology and chemistry and physics, but also the technology aspect. I mean, mentioning that it takes 40 years for someone to invent a laser so that Raman spectroscopy can be done, and then another 50 years for someone to come up with stimulated spectroscopy. How do you see the current wave of technological advancements moving at whatever pace they are moving at, and all of the excitement about AI and synthetic biology, whatever the next big thing in technology is? How do you see that coming to your work, and what kind of doors does it open?
Yes, this is, I think, a very exciting time right now to be in this field. It is very rapidly advancing. I should say that, my group, we focus more on applications where we're using the methodology to study particular biological problems. But there have been other groups, most notably Jishin Chen's group at Boston University, Wei Min's group at Columbia University, for example, who have been really pioneering the technology development to make it easier for researchers like myself to do stimulated Raman imaging. So yes, I think, from a hardware point of view, getting lasers that are much more reliable and less finicky has been really important. And so those lasers are now available commercially to researchers. Getting better detection systems so that you can measure these weak Raman scattering signals has been absolutely crucial, in particular for stimulated Raman, because it's not as straightforward to detect the scattered light as it would be with a conventional Raman system. You have to do a lot of electrical engineering tricks that you have to do, which a chemist would not necessarily know how to do. But, fortunately, the folks I just mentioned have figured this sort of stuff out. And so that technology is now becoming commercially available as well. I anticipate within the next 10 years, there's probably going to be commercial stimulated Raman setups that will be made available, that will be user-friendly enough that biologists could probably use. And then, you mentioned the software side of things. Once you get all this data, particularly if you're doing live cell imaging work, you're going to be collecting potentially terabytes worth of data. how do you make sense of that all? You can't individually hand process all that data. And so I think that's where automated approaches are going to become useful. There's been a lot of work from other researchers who are using machine learning techniques to denoise the data that's collected, to be able to help make sense of the data. So the data can often be very complicated and difficult to interpret. And so there are definitely algorithms that people are developing to try to help interpret the data. In terms of my research group, as chemists and not as engineers, we will certainly be using these technologies when they're made more accessible to help us answer questions in biology and chemistry. To recap, my research group is interested in helping to change the paradigm by which we do biological imaging fundamentally. It's not enough for us to know where a protein, for example, is in a cell at a particular point in time. We really want to understand what that protein is doing in the cell. We need to understand the chemistry. So we need to be able to understand how that protein is interacting with its local environment or carrying out, or what kinds of structural dynamics it's undergoing to carry out its function. And we believe that our Raman spectroscopy-based tools are amenable to getting that kind of information. So in particular, we're interested in developing these tools so that we can answer questions related to understanding the molecular basis of neurodegeneration, particularly for diseases like Alzheimer's. We're also interested in understanding fundamental molecular mechanisms behind which polymer and other types of nanoparticles deliver drugs and therapeutic nucleic acids to cells for cancer therapy applications. Then finally, we're interested in just understanding fundamental aspects about biology, most notably, how do structural dynamics of proteins differ and how are they regulated in the cell compared to test tube-based experiments, which is what most people tend to do.
For anyone who's listening to this episode, is there anything that you think they should do next after listening?
When people think about science, they always want to know what the payoff is. And sometimes with fundamental research, it's very hard to see what the payoff is, right? So we're trying to develop a new technique, and it's not quite clear that that technique always has an application, clinical settings, for example, or something like that. But I think it's important for people to realize that developing new technologies in science is vital towards advancing science and making breakthroughs. The microscope and the telescope, I would argue, were two of the most important developments in science because it allowed us to study things that we could never have seen before. Things on the very small scale, like cells, and things on the very large scale, like planets and galaxies, right? And had those technologies not been developed, we would not know about a lot of our universe, right? And so in that same vein, there are researchers like myself who are trying to develop new imaging tools that can allow us to probe new areas of science. So people can go to my website to find out more information about my research. Go to thepunihaolegroup.squarespace.com.
Dr. Punihaole, thank you so much for your time and hope to speak with you again sometime soon.
Thank you. Bye.